thank you for taking the time to speak with us today regarding your article published in Thrombosis. If you could start by just introducing yourself and perhaps summarizing a few key points in this article.
My name is Ofosu from McMaster University. I've had the pleasure spending about 2 years to write this review, to get it right. The points that I tried to get across are the following: that low molecular heparins are very good drugs, well-established properties and safety and so on. So now that many of them have gone off patent, it's useful to have non-proprietary versions of these low molecular heparins worldwide. In some countries, they're known as generic low molecular heparins because in those countries, low molecular heparins are seen as drugs. In other countries, they are known as biosimilar low molecular heparins, simply because low molecular heparins and heparins are seen as biologic medicines in those countries. Now, this distinction creates a lot of headaches, especially for people who make low molecular heparins, because there are different rules for regulating them, and it's tough to make the same manufacture one piece of medication and have it regulated in 2 different ways in 2 different countries. So this is the biggest headache with 2 ways of looking at these medicines, one as drug and one as a biologic. The time has come for people to wrap their heads around the issues and then come up with a simple, a singular way of looking at these substances throughout the world. So it's a bit easier for manufacturers to make them, and also patients and the doctors to be more comfortable knowing that you're dealing with the same substance. These were the points that I was trying to make in the article.
What would you say some of the reasons behind the very different regulatory positions of the FDA and EMA regarding non-proprietary low molecular weight heparins?
Well, the big difference, frankly, is how they are viewed. If it's a drug, at least in the US, and also worldwide, there's a fairly straightforward way of making copies of drugs. You just have to make sure the structures are identical. Once they're identical, then it can be used interchangeably. You don't have to do any clinical trials to use drugs or copies of drugs that have been manufactured to the same specification. But if they are biologic, the thing about biologic is that, a drug can be synthesized in a fairly straightforward, well-defined chemical steps. Biologics usually are not synthesized outside of a living organism. You need a living organism to make it. Yeah, drugs are easy to manufacture because they have well-defined steps for synthesizing them from simple chemicals. Biologics, on the other hand, are not synthesized outside of a living organism. So a biologic can come out from an animal, such as a heparin on it, or insulin, or it can be isolated from plasma, human plasma, sometimes animal plasma, or can be made by recombinant DNA technology. Doesn't matter which way a biologic is made. Not all the molecules are identical. For all kinds of reasons, you really don't, there are a series of components in there, some of which may be very active, some of which may not be active. So you really don't know. You have a population of molecules with varying properties. So it's extremely difficult when 2 different companies make the same product using a biological synthesis for you to be sure that they've got an identical product. So in fact, all you can be sure of is that they are similar. The only way you can be sure that they are effective is to try it first, perhaps in animal models and also in patients, to be sure that they work, and they work as you expect. and also they are safe. So making copies of biologics really means that you're going to have to do some clinical trials. Whilst if it's a copy of a drug, generally no clinical trials are necessary. So this is where the big headache comes from. In some countries where the same product have to be tested in patients before they can be approved because it's viewed as a biologic, whilst in other countries the same product or something similar to it does not have to be tested in patients at all because it's a drug. So it's a philosophical question. It's a tough question.
What methods are available to agencies like the FDA to determine whether or not those drugs have the same properties as the parent drug?
Well, from the FDA's perspective, because it's a drug, generally they don't require any studies at all in patients. But an exception was made for low molecular heparins, because all of us recognize that even if it's a drug, it's not a simple drug. And nobody knows precisely which constituent is required for activity. So the compromise that the FDA made was to ask the manufacturers to show information that the drug, in fact, behaves like the comparator drug in patients, at least one is given to healthy volunteers, that you recover the same amount of activities and so on and so forth. So this clearly is beyond what is usually required of a drug. But even if it's a drug, it's a complicated drug. So FDA required some sort of in vivo evaluation. And they were satisfied with the in vivo evaluation. But also, the FDA required all kinds of very detailed information about the structure and the composition, simply because one cannot be just by measuring one aspect of the heparin come to a conclusion that it's exactly what it is that you're looking for. So it took a long time, to be fair to the FDA, it took a long time, several years actually, before they approved the 2 low molecular heparins approved as generics in the US. I don't know exactly how they did it in countries where the generic low molecular heparins, such as Argentina, China, India, and Brazil, because they haven't told us how it was done. But unlike the United States, some of these low molecular heparins have been withdrawn from the market in Argentina, India, and China because they were found to be unsafe. Given when these withdrawals took place, I'm not sure whether this was because of the contaminated heparin components may have been in there as well. It's possible, but we don't know because they have not given us that information.
One of the next questions I had for you was also regarding the manufacturing companies, and with all those requirements, how can these agents be required to prove that the low molecular weight heparins have comparable safety and efficacy as their parent drugs?
The easiest way to do it, and this is where the assumption is that they are biologicals. The way you would do that in that case is also to do safety studies in healthy volunteers and do efficacy studies in patients to look at them, to sort of be sure that they what you think they are. For a biologic, you need to do some studies in healthy volunteers as well as in patients. Healthy volunteers, basically to establish that the dose will be safe, you do not have any issues in terms of safety, and efficacy in patients who have, who need the drug, such as low molecular heparins, because they've had surgery and they are at risk for developing blood clots. There are two things that the manufacturer needs to show. The safety would usually be looked at in the first instance in healthy volunteers, so that when it's given to healthy volunteers, the drug will be found in their body as predicted. But the efficacy and sometimes safety as well, is determined in patients who require the drug for a specific indication. And then you have evidence that the drug works and it didn't cause any unexpected events, safety events. So it's a little bit harder. It requires, obviously it's a lot more expensive doing these clinical trials compared to generic drugs where no clinical trials are required. So this is the big difference between Europe and the United States at this stage. Now, to be fair to the Europeans, they have evaluated a whole lot of biosimilar medicines and approved them since about 2005, where clinical trials were done as is required for low molecular heparin. We might ask ourselves, why is it that in Europe, especially when it comes to these biological, especially recombinant biologicals, why they will do these trials? Well, I think one reason is that heparin is fairly inexpensive compared to any biological, any recombinant therapeutic protein. Those are fairly expensive. So there's a bit of a room for somebody making copies to, in fact, be prepared to spend the money to do the clinical trials, because they can sell it for a lot more money than they can sell low molecular heparin for. So if you buy that premise, then personally I'm not surprised that no manufacturer in Europe has made a biosimilar low molecular heparin, because chances are after the clinical trials and so on and so forth, there probably will not be a whole lot of profit to be made if they spend amount of money to do large clinical trials. This clearly is not an acceptable situation, because these drugs will never be licensed in Europe. If you wrap our heads around it, clearly these generic low molecular heparins have been approved in the United States. The question is, are they safe and effective? I have not read anywhere that they are not safe and effective. In fact, they've been used for almost 2 years in the United States. And that's a fairly large market. So that if there were some safety issues, they would have come up quite some time ago.
Would you say that Europe and the US are moving in the right direction when it comes to regulations being synonymous?
In some areas, the answer is definitely. If you look at vaccines, for instance, pretty much the same rules are in place in Europe, the US, and the rest of the world. Why is that so? Well, because these things are done because of the huge public health issues with vaccines. They are done, the regulations are run, at least written, under the auspices of the World Health Organization, to which most countries make important contributions in terms of regulators from several countries are involved, manufacturers are involved, so that it's easy to come up with a consensus as to how to do these things. And the same drug made in Europe or United States can be sold almost anywhere, because they were manufactured under the same set of rules. Okay. And obviously vaccines are biologicals. So there's a general agreement as to how biologicals should be looked at. Aspirin and other synthetic drugs are clearly drugs, and again there are universal rules as to how they should be done. So as long as there's an agreement as to what the agent is, it's fairly straightforward to write regulations that most people will find acceptable, because everybody contributes to a consensus development and makes it easier for any manufacturer who meets the requirements to be able to sell their products in anywhere in the world. This is a situation where clearly half the world is looking at it as biologics, half the world is looking at it as drugs. So there's no consensus. This is where I'm thinking that we need to really wrap our heads around it, and regardless of why we call it, figure out how best to make the substance safe and effective in ways that most people agree that is safe and effective, so that it will be more readily available.
I guess going back to interchangeability and comparing low molecular weight heparins and their parent drugs, what are potential drawbacks in assuming that the interchangeability of non-proprietary low molecular weight heparins and their parent drugs?
There are two answers to this. In the first instance, if you take a look at the low molecular heparins, the original low molecular heparins that were approved, each is viewed to be a different drug, so it cannot be used interchangeably. In the first instance, if you take a look at the low molecular heparins, the original low molecular heparins that were approved, each is viewed to be a different drug, so it cannot be used interchangeably. Low molecular heparins that have been approved in Europe and United States, the originator ones, are each seen as a separate drug, so they cannot be used interchangeably. Now, you need to ask yourself, why are these 8 low molecular heparins viewed to be different entities? If the answer is that they are different entities based on fact, and I really don't know the basis why the regulatory authorities in both Europe and the United States decided they were different. Is it possible that the manufacturers made a very strong case that they were different, and perhaps in reality they are not? If the minor differences between them, chemical differences between them, really are significant, then I think we have a big issue to resolve when it comes to the generic or biosimilar low molecular heparin. The only way they can be deemed to be identical, at least interchangeable, is if studies have been done where you have used the original and a copy in the same person, and you got exactly the same effect, and in fact back and forth. Until you've done that, you cannot say that they're interchangeable. If, on the other hand, the differences are really not important for the existing approved 8 low molecular heparins, and they can be used interchangeably, then frankly, the copies that are made for any low molecular heparins could be used in exactly the same way, and perhaps even all low molecular heparins could be used interchangeably. I really don't know the answer, because we don't know. The studies have simply not been done, which led to the determination that they were different, and no studies have been done to show that in fact they could be used interchangeably. So it's going to be a tough one. And again, it may very well be that a consensus will be built up, and people may have to do collaborative clinical study, both in Europe and United States or worldwide, to gather the information. And these are important concepts. If the drugs are not interchangeable, they should not be used in an interchangeable way. But if they are, so they should. Frankly, it would be a lot easier if low molecular heparins were deemed to be interchangeable because studies have been done to show that they are interchangeable. We need information that will confirm to us that any 2 low molecular heparins that are now deemed to be different drugs can be used in the same indication safely. If that is indeed shown, then I think it's fair enough to conclude that copies of other low molecular heparins will be used in an interchangeable manner. But the only way we can get that information, I think, is that the studies have to be done. Low molecular heparins are used quite extensively, so that if regulatory authorities insisted that this information should be gotten, it will be gotten. It's going to have to come from the regulatory authorities, in this case, not from the manufacturers. The manufacturers have no interest in. It's a lot more reasonable for them to assume the position that the drugs are different from the other guy's drugs. But regulators don't have that concern. They want to be sure that they can regulate the drugs appropriately. But if they're the same, they should not be marketed as different drugs. You see my point? And only the regulators are in the position to ask for these sorts of studies to be done.
How does your article relate to the North American Thrombosis Forum and our members?
Clearly, North American Thrombosis Forum has a big interest in advocating the appropriate use of antithrombotic drugs to prevent venous thromboembolism, an important clinical entity which doesn't get very much attention, get as much attention as it needs. Now, low molecular heparins are important drugs in the use. They're used to prevent and to treat thrombosis, both arterial and venous. So the more of these drugs that become available, chances are the less expensive they might be, especially if copies could be made safely and that are also effective. So I think the forum has a real clear interest that these drugs become more widely available and they are affordable. So competition is the only way to drive the prices of things down. But I'm sure also that low molecular heparins are not the only drugs for treating or preventing thrombosis. As you are aware, a whole set of direct factor Xa inhibitors and direct thrombin inhibitors have also become available. And frankly, as they be used more often, this may very well drive further the price of low molecular heparin. So my hope is that the field, will continue to grow and prosper, because the more drugs that become available, the more likely that prices, especially for the older family of drugs, will go down. That will only be beneficial to patients as well as the people who are paying for the drugs, insurance companies, and so on.
What do you want readers to take from this article?
Basically, that these low molecular heparins are good drugs and they will continue to be good drugs. They may be difficult to regulate in Europe and the United States, especially in Europe, because of the difficulties that clinical trials entail, especially the cost. So there's something to be said for perhaps Europe learning from the American experience in terms of going to regulate these drugs and find out whether or not there can be some cooperation to regulating low molecular heparins. So the people who manufacture them in Europe are going to be able to sell them in the United States and vice versa, and also elsewhere in the world. Because if there's one way of regulating low molecular heparins, then pretty much any country will be comfortable purchasing the drug, because they know it will be the same drug whether it's used in Europe or in the US or in China or in Africa and so on. It will be good to find some sort of consensus as to how to manufacture and regulate low molecular heparins, just as we done for vaccines and biologicals.
I think you answered all of our questions over here, but on behalf of the North American Thrombosis Forum, Dr. Goldhaber, Dr. Fareed, our staff here, we want to thank you so much for taking time out of your day for speaking with me today.
It's a pleasure, and I certainly enjoyed your questions. There are interesting questions that need to be addressed, so I hope I've been able to answer them in a way that makes sense to listeners down the road.